Is Dr. Jeremy Schmutz is a faculty investigator at HudsonAlpha. And Jeremy is one of our very first ever customers. Was it 2010 or 11? I think you were certainly in the top 5 of ever receiving an RS instrument back in the day. And he's just been a great champion and user of our technology. And will tell us about the latest that he's been doing. We're honored to have Jeremy. Thank you.
Thanks, Jonas. I think we all need to wake up a little bit here, because it does feel like day 973 of PAG at this point. So anyway, we're going to talk about this. And the right side there is a very nice picture. I drew it myself of a cornucopia. This is apparently what a cornucopia, universal cornucopia for genomics looks like. I'm going to talk a little bit about project coming from both my JGI work and from the HudsonAlpha Institute. First beep in the past, in Science in 2010, where we had very large monitors apparently. Still, there's a quote there, the most promising sequencing advance has been the development and commercialization of single molecule sequencing for specific biosciences. It can produce 2,200 base pair sequences. However, at this point, the accuracy of the reads is unknown, the throughput is also unknown, and we don't have any evidence to suggest this instrument or sequencing methodology will be directly useful for a pine genome. That's from a grant. Maybe those people are actually working on strobe sequencing or direct RNA sequencing. I hope PacBio is still working on them and will eventually release those products. Okay, so this is why HiFi is cool and important and has changed really the way that we think about working with plant genomics, human genomics, animal genomics. The left side is that long read versus HiFi, and you can see the difference really apparent down in alignments from there. And what that's really done for us in the HiFi world is it's changed the way we do computation and really sped up all of the things that we can do with that, because we can do direct matches as opposed to this thing that used to happen with long reads, which we don't want to talk about anymore. So just to give you a simple example of one of the first outbred genomes that we sequenced and published is the CJ Size work on Populus. There's a hybrid. Christine, it's a hybrid, in case you're interested. And CJ's ended up going in here and using this really to identify and knock out all the trichome genes, which turn out to be variable copy number depending on the haplotype. So years and years they tried to knock these things out, and now having a haplotype-resolved reference enabled them to be able to precisely do that. And I'm going to say that that's just a small genome, tiny poplar genome, and recently been working on quite a few pine genomes, and there's soon to be a dime a dozen. That does not mean they will actually cost a dime if we actually have dimes anymore. However, there will be many gymnosperms coming out in the next couple of years, 63 times bigger than poplar, beautiful contiguity, better than what's been achieved previously with long reads, which is not that surprising. And then just to mention sugarcane, because for some reason I'm required to mention sugarcane in every single talk about PacBio, is that the R570 genome and paper came out, and it is really amazing. We were able to resolve at least 6 to 8 copies across the sugarcane genome there. And it's really beautiful, nice, and it provides a lot of discussion right now about moving on sugarcane genomics and really starting to do that for real relative to what's been done in the past for that. Today, I think we can really redo this again just with OmniC and HiFi and our normal pipeline. Back then, we did a lot of other things to produce data. So just a bit of what we're doing with PacBio. It's a better tool for disease discovery, plant pangenomes, improved annotations, what I call devious dirty microbes, and then for the final segment of, wait, what, there is another PacBiochemistry. Inconceivable. Anyway, we've been working on rare and human diseases for a while. We switched everything over to PacBio HiFi sequencing now to identify rare disease, led by Greg Cooper as projects, and we've consolidated all these cases for many years together and have been putting them into 30x HiFi genomes and assemblies to try to improve diagnosis. The goal is to move these no returnables on the left all the way up into identifying likely pathogenic and pathogenic causes of these diseases. And there's a paper in Genome Research to talk about that, and just to show you one simple example of that. This is a 4 megabase inversion. It's very difficult to call from short reads, if you can believe that, and it's pretty impossible to call from exome capture. There's nothing there. And we're able to not only just call it with a HiFi, but also build out the assembly and validate it, validate both copy numbers. That's just an example of looking at as we move things from uncertain into diagnostic, likely diagnostic cases. So we're still got quite a few that we can identify, but can't classify and don't know that they're really linked to the disease. And that's the dark matter, if you could think about that, of human disease. And you'll see it again and again in everything that's talked about with PacBio, because all we're doing now feels like is discovering new dark matter that we didn't really know existed before, and then we have to figure out what it does. So just to talk a little bit about pangenomes and improved annotations, this is just a couple examples of pangenomes that are ongoing or in publications that we've generated, and just to fun lots of different plots. Some of these are, many of these are gene space plots. We've really scaled up. That's one advantage here in the last couple of years of HiFi, to get to the point where we can produce 30, 50, 60, 100, 400 genomes now of species. And so just to give you an example of recent sorghum, which is in bioRxiv now. That's a diversity collection of sorghum, in case you haven't seen sorghum before. This is a project that was originally started by Todd Mockler and led across the years by Jeff Morris and Nadi Shakur, and that's to classify all of the world's diversity in sorghum, and we've really ended up trying to generate that and put that into this diverse pangenome that crosses all over sorghum, a diversity, domesticated diversity. And you see on the bottom there just some pangenome graphs to show what we were just talking about a little ago, is that as you add pangenomes, add new genomes to your pangenome, you also add new genes, and those expand along the way. This is an example of the dhurrin biosynthetic cluster. Dhurrin is essentially, you can think of it as a pesticide that gets generated by sorghum to reduce herbivory, and it also is associated with drought tolerance. And there's a nice BGC cluster of it on chromosome 1 with some variants in SNP calls, and that's an association study. When we turn this around and look at it in the pangenome view instead, what we see is that there is additional deletions that we do not see with short reads, and that are not necessarily linked to SNPs. And we also see multiple new haplotypes captured there in that nice graph. And then we can map those back into the original populations using K-mer-based methodologies and really track the sort of evolution and history of that. And in this case, you see some of the association of specific haplotypes with the specific regions that are under drought or increased drought stress across Africa. And there is a poster 653 to check that out. So my dream for 2015, well, I guess from 2015, was to have a sorghum genome. Honestly, that's all I wanted, was a high-quality genome. And I thought, well, wouldn't it be cool if we started looking at more than one genotype? And so we started looking at more than one genotypes. And I thought, let's add some more genotypes and some resequencing data and get some variants and some SNPs and all that stuff. And that's really the pangenome of domesticated sorghum. And that's kind of where we are right now. And we can make it better and we can add more to it. But we're capturing large amounts of diversity that are available. And then I thought, well, wouldn't it be even cooler, though, if we could look at sorghum diversity that includes the wild sorghums? And that's really the pangenome of the genus sorghum. And so in a project led by Robert Henry and Rosalind and Blake, we've been working in the wild sorghums. And these wild sorghums are all in Australia, and they're all really small, weird-looking plants. They don't look anything like the sorghum I showed you earlier in the domesticated sorghum germplasm collection. But what crazy diversity we have here in sorghum. You can look at some of the stats, but we're producing these amazing genomes which are resolving outbreds, multiple haplotypes, 4Xs, and you can see at the top that bicolor. That's our tiny little one that we just generated, 30 genomes in the pangenome, and now at the bottom there you can see that gigantic 8 gigabase monster there, which is all, well, anyway, all still related. Check back in and see when we figure out how to do biology with these. Okay, so this has all led to our greatest problem that we've had in the last 3 years now, which is what I call pangenome annotation woes. So in some work that's led by Tomasz Bruna and John Lovell in my group, they've been looking at this question of how do you stop the proliferation of bad genes in pangenome annotations. And this is just an example of taking all the cotton that had been annotated and available in the world at the time, and then re-annotating it with our internal pipelines from the JGI. The blue there is the constant expansion of genes going on when you add new genomes, and then the red shows that we do expand, but we don't expand as fast as a rate there. So there's the external annotations there, have an enormous number of these 1 copy, 2 copy, 3 copies there. And with the JGI annotations, we at least get rid of some of that variation across the genomes. However, we'd like to get rid of more of that, going back to what was just brought up earlier today. And so what we're doing now is develop this panJGC methodology to predict genes in individual members in a consistent manner. So that's with IGC pipeline, and that uses full-length cDNAs, isoseq, and Kinnex plus Illumina RNA-seq to be able to do that. And that's kind of what individual annotations look like. And then we go back through, map all the predictions across the pangenome, update them all to maximize a function that optimizes consistency across the pangenomes and support by extrinsic evidence. Thank you. That's an EE. I'll just call it EE for now. Okay, so that's what they look like when you've updated them into there. And just to show you a quick example of this, this is a cluster, which means that this is very confusing for annotation because you drag your resources out over multiple copies and then try to make sense of them there. And you can see what the sorghum pangenome looks like. We've got a kind of a nasty looking ones where we've predicted really weird stuff in this gene cluster. And that's what the transcripts look like from one of these worst ones, where we've taken this nice looking cluster, which I could just look at and say, hey, I know where the genes are, and then we've smashed it all together in weird ways. The new panJGC predictions then cleans up that, fixes the UTRs, and gives us a really nice consistent annotation. And kind of what that looks like is, and this is a relatively simple example, we can go from the left side to the right side, and we can increase the number of gene clusters which are identical, decrease the number which are rare, and while still maintaining consistency across all the variants in the genomes. And so we don't lose any information here. We're only making the information that we know is accurate a little bit better and making it way cleaner to be able to do anything with these gene sets. Here's our example that's taken a long time to work on, which is part of the reason we're still working on this, because Eucalyptus grandis has massive numbers of tandem gene duplications, and they're very difficult actually to resolve when you start asking questions across the pangenome because they're large-scale PUVs. But that's what we want, is we want to know which ones are there in present or absence. And so that just shows you a nice gene set. The original gene set and the panIGC G set, where we restored something like about 4,000 genes to that. But in the process, made this a whole lot more usable for everything we want to do with it. Finally, we have annotations for pangenomes that we can actually accurately compare gene content. Okay, so then I'm going to talk a little bit about dirty, devious microbes. They are microbes that live in dirt. And this is a project led by Tom Younger and Joe Edwards on switchgrass. Everyone has heard me talk about switchgrass many times, but this is actually doing cores and then doing a 16S to try to understand the populations around the cores. What we've been doing is collecting these cores and then collecting these dirt cores in order to be able to make cores of microbiome for each one of these roots. We use a bioprospector to increase our enrichments, and then we sequence the core microbiomes to build this core throughout the root associated microbes, the rhizoplane, and also the endosphere. And so this is kind of what it looks like. We've got 3,067 isolates that we've made, and we sequence, and that's kind of all we're trying to do really is link that to the original 16S data from there. One of the problems with that, though, is that 50 species, for example, of actinobacteria have exactly the same V4 sequences. If you don't work in root and you work in a human microbiome and you kind of only care about the genus, then this stuff isn't that important. But if you actually want to know who your individual community members are and how they're associating themselves with your plant, it becomes key to be able to tell these apart. So we've deployed this, the 16X, day 698. We've deployed this, the Kinnex version of long 16S, and have really been able to sort of try to figure out how to move this project forward. And we've been able to show really nice stuff, like we can tell the difference between sites, we can tell the difference between the 3 compartments, and they look really nice and resolve well. And then we've also been able to get over this problem, which is an example of looking at all those V4 sequences now, and you can see that by the size of the circle, that many of them are things that are completely hidden when you're looking at a short V4s or other approaches on 16S. However, just to go to the dirty, dark, devious microbe discussion again, and dark matter, only 16% of these actually match to anything we can find. So there is somewhere in the 84% range of completely new sequences when you look at the full region for these microbes. So another place to do a little bit more work to figure out what's this new stuff that we're finding and what it is. And dirty, stupid microbes. Okay, anyway, Joe has a long read sequencing and plant microbiome research for Tuesday at 6:20 p.m. That is an awesome time. Almost as good as my time, which is 6:20 p.m. tonight for VER Plant Genomic Science here, if you're interested in hearing some more about these things. And I just want to thank the users and partners in DOE projects, because a lot of this has come from all of our work over the last 18 or so years in building plant genomics out for the world. Okay, so wait, what? There is another PAC biochemistry. Inconceivable. Okay, if you're like me, well, you just finished transitioning to the previous chemistry. Maybe you're not like me and you transitioned January 3rd of last year, but we have not. We did not. We've been transitioning, but it's really slow. So now we've got a whole new chemistry, and along with that is this new SPRQ-Nx, if you'd like me to pronounce it that way, SPRQ-Nx chemistry, which brings the highlights from, although rearranged on Jake's slides, is this multi-use smart cells and the chemistry upgrade and what it brings to you. So this is what it is. So it's the experimental group led by Jane and Melissa, and we've been part of the beta now for a bit more than a month and a half, almost a month and a half, but there was like a bunch of that was busy doing other things like generating AI pictures of cornucopias. So what do I want to know? I want to know what the yield and base pair accuracy is. I want to know about that second chip and what does it do and whether there's yield loss and fall off on the accuracy, is there carryover between the runs. What's the accuracy for SNP and SV calls, and can we build accurate genomes out of SPRQ-Nx? Okay, so what's the yield and base pair accuracy? You'll see these a lot, but essentially these are what we use for controls. We use a human control. We use a reasonably similar GC content, usually sorghum control. We use a bean, which is a low AT with lots of interesting sequence patterns in it. And then we use a poplar tree, which is our outbred AT-rich control. So in this case, if you just look at here, you'll see that for the most part, SPRQ-Nx is pretty good at generating gigabases. In the human case, we see a pretty reliable increase in yield, and you can see it goes down a little bit in sorghum, goes up a little bit in bean, and comes down in the middle somewhere in poplar from the previous chemistries. These are all one chip, so it's a little, not that informative. What's the yield and base pair accuracy? We borrowed some data from PacBio for the hydidiform mole, and for everybody's benefit, I did not put a picture of a hydidiform mole there. No, okay, sorry. So yeah, this is great because this is a single haplotype, and so it's really good at reliably predicting. We use Google's best algorithm, which they developed to be able to test and develop the original deep consensus, and shows something like around similar, a little bit actually improved quality in SPRQ-Nx. Sorghum, which is again an inbred, you can see also similar, and remember it's the same about GC range as human, quite close to, getting pretty close to 1 in 1,000 errors roughly from there. On the AT-rich bean, which also has a lot of sequence motifs, you can see it's fairly consistent. Here I'm showing it a little bit lower quality than the V13, but fairly consistent there at closer to 1 in 100 than 1 in 1,000. And that's just that's the wonders of this. Poplar goes up a little bit. Poplar has a little more complex sequence than bean does, and so that's where bean tends to accumulate the errors. How about yield loss? Yield loss. So yield loss, we got a little lower on the second one in human, a little lower in sorghum, a little bit higher on bean, and a little bit higher on poplar. So it's essentially, if you've never ran the same library multiple times on PacBio, that's about the variation that you see anyway. So it's really interesting to see what that will continue to evolve in, and we have run a bunch more samples, and it's pretty similar. It's kind of like this variable. So how about accuracy fall off? So now if you look at, this is just QVs here, you can see the similar kind of pattern, a little bit lower, a little bit higher, a little bit lower, a little bit lower on the total reads that pass filter here for that. So that seems all pretty consistent. When we look at carryover between runs, we vary a little bit, but I think the target is like 1 in 8,000 or so. We get that 1 in 10,000 and 1 in 12,000. We do have a higher carryover when we first load a diatom, which is a short, generally crappy, low-size library on, and then load human afterwards. However, when we went back through, we did verify that we're actually, with the existing system, we are not actually moving any reads from diatom to human and vice versa. So it actually seems very clean for moving between samples. Hopefully that turns out to be, some, I'm going to call it, enhanced washing needed around some short reads. This is unsupported at this point in time, and we'll see how that goes later on. Just to show some PacBio SVs, this is on human. Again, we run the same human sample. We downsampled, everything's the same. The only difference really is we're getting a little bit less break-ends, which is actually pretty good because most of the break-ends are totally bullshit garbage anyway. They don't really tell you anything that's going on there. So that looks really nice. And then looking at SNP content, we're actually a little closer on our SNP calls back to V13 relative to SPRQ data. So it looks pretty clean, and there's good consistency on there. Okay, so building contig genomes. I don't know, contig genomes? What was I thinking? So we're building genomes, but we're just building contigs. Just to evaluate the question of the chemistry. The humans are always 25x because that's what we use in our diagnosis. And then the sorghum, bean, and poplar are all 40 to 50x per haplotype because that's what we use when we build genomes. So if you look at that, there's 2 represented for human and 2 for poplar. Generally, the amount of coverage is pretty consistent, or so the amount of sequence assembled is pretty consistent. The contig N50 is pretty consistent. In several cases, we're doing a little bit better in the SPRQ-Nx than we were in SPRQ or V13. And then here's the Buscos. The thing you might notice on the Buscos is it's pretty consistent for human, pretty consistent for bean. Sorghum is a little bit low, and we're getting some mixed signal on the poplar. And so we have been looking at this, and when you go back and you look at hap1, hap2, okay, let's just try to back up 1 second. The goal here is to generate a single haplotype of sorghum, and we do that. But when we look at a single haplotype of sorghum, it only has about 97% Busco rate instead of 99.1. And the reason for that is that HiFi ASM is doing its old funness, and it's, even though you want 1 genome out, it's still giving you 2, and it's not very good at necessarily assorting it. And so there might be a little bit of work that needs to go back in there, and if you're going to build contig-only assemblies to be able to figure out whether you really have a hap1 and a hap2, or how to generate your single haplotype reference. You can, of course, fix that the way we do all the time and have forever, which is put OmniC into that, and then you can get back, recover the Buscos and the contig N50 out of that and produce really nice looking genomes. So, okay, current thoughts. Mostly it's a drop-in replacement for SPRQ with comparable quality as far as we can tell. I know it seems super boring, sorry guys. Aren't you glad you waited this long? Lower projected cost per gigabase, although we do the reuse of cells, although I don't, all I have is the PacBio marketing numbers for that, so I don't know what that will turn into for things like plants or other exciting things we work on. Slightly higher HiFi yields, I think it looks like, looking at our extended data sets coming out of SPRQ-Nx versus SPRQ on there. There's some downsides. It is kind of complicated in the laboratory because it does require 8 cells worth of libraries to load there, and so when you, it's going to end up being a little bit different. It's not so bad if you're doing a large resequencing project with huge numbers of cells, because that makes it fun and easy to load. But if you're actually, loading smaller things like a plant genome, for example, then you're going to have to mix it up a little bit and figure out how you're going to do that. Also, you may need to optimize your existing assemblies or visit some of your pipelines, particularly for the inbreds. We didn't see any. You saw pretty clean separation and niceness on the outbreds. So I work as part of the JGI and lead the plant program for that. We do have a excuse me, something called a community science program that you can apply to. If you have a DOE relevant program, a lot of these are coming out of that program. We also have a user meeting, which is going on in a few weeks here. And you're welcome to join us in Seattle for that. All of our data is available on Phytozome as we produce it. And so that's this plant thing where you can use to compare things on the site. And so you can go there if you're looking for some of that data. And I also invite you to come genome with our team at HudsonAlpha. On the top there is some of our experimental experiments we're working on, protocols from our experimental group, including I had to put one fiber-seq picture in there. I think it will be incredibly useful and great, but I think it's not quite as easy as Christine makes it out to seem.
Computational analysis.
Right, yeah, the analysis is great. To get to that point from plant tissue is sometimes a little bit harder. And then also our computational analysis group, which builds these plant genomes and integrates all of that analysis. And so feel free to email me, find me, and we can collaborate on the sort of future of genomics. Additional contributors from the JGI production group, or Greg Cooper's group I highlighted. We have a plant functionals group. Arizona Genomics is our partner group that does a lot of RNA and DNA extractions for all of these projects. Phytozome and annotation group, which I talked a little bit about the work they're doing in computational genomics group, and our project management office led by Carrie. Okay, thank you. That's it.